Now, you know that your DNA means deoxyribonucleic acid. Okay, so we say this is very important because this is the molecule of heredity. Okay, this is your genetic information. This is the genetic material that can be found in the nucleus of our cells. Okay, so your characteristics, especially your physical characteristics, even those that we can't see, your genes are dictated by your DNA. Okay, your DNA structure, and of course, these are coming from your parents. Now, for organisms to grow and repair damaged cells, each cell must be capable of accurately copying itself, or this is what we call DNA replication. It's very important that your cell is able to replicate or to make copies of the DNA, okay, because it is very important for your growth. It's very important for the repair of damaged cells. Okay, so again, you were asked to watch a video about the structure, the function, and history of DNA. We are going to be talking about the different things that you've watched in that video. Okay, we start with the history of DNA. Now, the very first person who've learned about DNA and its very important characteristic being your hereditary material was Frederick Griffith, and this happened in 1928. Okay, so that's the first person to have studied the most important characteristic of your DNA as your genetic information. Okay, so that's Frederick Griffith. Again, that was in 1928. Now, what happened was that he discovered that heat-killed virulent bacterial cells release hereditary material that can be transferred to harmless cells, making them harmful. Okay, so what he had was he had some virulent bacterial cells and he had some non-virulent bacterial cells. Now, again, when you say virulent, that means harmful. Okay. Now, when he killed these samples of his bacterial cells, he found out that they can no longer kill the mouse. Okay. He used mice in his experiment. So this was his experiment. At first, he used smooth strain. These are your virulent strains of bacteria. Okay. So when he injected this to the mice died. Okay. So again, your smooth strain bacteria are virulent. Now, he also used rough strain bacteria, another version of your bacteria, but these are non-virulent. So when he injected this into the mouse lived. Okay, so the mouse still lived because, of course, your rough strain bacteria are non-virulent. Now, what he did was he killed his smooth strain bacteria using heat. Okay, that's what we mean by heat-killed smooth strain bacteria. So again, he killed his smooth strain bacteria using heat. And so when he injected this to the mouse lived because, of course, the bacteria were already dead. Okay, they were killed using heat. Now, what he did in his experiment is that he combined the rough strain non-virulent bacteria, which is of course living, with the heat-killed smooth strain bacteria. Again, remember, your heat-killed, these are dead bacteria. While your rough strain bacteria are living bacteria, but these are non-virulent. Okay, this does not cause any harm if injected separately into your mice. But what happened was that when he tried to combine these two, the heat-killed smooth strain bacteria and your rough strain bacteria, which is non-virulent, both of them, when he combined it, the mouse died. Okay, and so that led him to conclude that the transfer of genetic material from one cell or one organism would have DNA involved. Okay, so he said DNA, there's something. He actually did not pinpoint it to be DNA yet. Okay, he just said there's some hereditary material, there's some material in that bacterial cell that even if it has been heat killed, it still was able to cause harm into these mice. Okay, so he said he called this transfer of genetic material from one cell or from one organism to another cell or organism as transformation. Okay, so that's transformation. But then again, we need to remember that Frederick Griffith was not sure whether that part of your cell was DNA, whether that part was protein, whether that part was RNA. He was just sure that there was some sort of hereditary material inside that cell that was transferred from one organism to another. Okay, so that was Frederick Griffith. Again, he said there's some parts of your hereditary material, some parts of the cell that was able to transfer into another organism, to another cell, and that caused the mice to die. Okay, so again, you need to remember, he was not sure what type of material that was. Okay, he just knew that there was some hereditary material inside the cell. And so the next person to continue the study was Oswald Avery. Okay, and this happened in 1940s. He wanted to determine if protein or DNA or RNA was transferred in Griffith's experiments. Remember, we said Griffith was not sure whether it was protein, whether it was DNA, or whether it was RNA that was transferred during his experiment. Okay, now what Oswald Avery has discovered was that it was actually DNA that was responsible for transformation. Okay, it's not protein, it's not RNA, but was DNA. Okay, let's take a look at this experiment. So this was the experiment of Avery, and his hypothesis was that the genetic material of the cell is either protein or nucleic acid. It can either be DNA or RNA. Okay, so he got some heat-killed S bacteria. Okay, these are the smooth strain of your bacteria. And he added some lipid, he took out some lipids and sugars, okay, because he did not need this in his experiment. He only had protein, he only had RNA, and he only had DNA, okay? Now, what happens is that when he did this and he tried to check the results, okay, the resulting strains of bacteria that you have here, there is an S cell that appeared here. There is an S cell that also appeared in your second experimental setup, but there is no S cell that appeared in the third experimental setup. Okay, so even if there is no protein in this concoction, there is still an S cell that appeared. Even if there is no RNA in this vial or this test tube here, there is still an S cell that appeared. But if you take out that DNA, okay, if you take out the DNA, then there's not going to be any S cells that would appear. Okay, and so he concluded that it is actually your DNA transformation requires DNA. Therefore, the genetic information, genetic material of the cell is actually DNA. Okay, so Oswald Avery was able to pinpoint that genetic information is transferred by DNA, not by protein and not by RNA. Okay, so that's a breakthrough in the study of heredity during his time. Now, another experiment was done by these two very important people. There's Hershey and Chase. Okay, so there's Martha Chase and Alfred Hershey. They confirmed that DNA is the hereditary material used in bacteriophages. Now, when you say bacteriophages, these are bacteria. Okay, so when you say bacteriophages, again, these are viruses that can infect bacteria, and they use this in their experiment. Okay, so this was the experiment of Hershey and Chase. They used, in one type of their bacteriophage, they use proteins. In the other type of bacteriophage, they use DNA. Okay, and they also used their radioactive material so that they can see whether there's some radioactivity inside the cell once it has been injected by protein, or whether there's going to be some radioactivity inside that cell, the bacterial cell, if they inject DNA inside it. Okay, so they injected both protein and DNA. Okay, so your phages, your bacteriophages, your viruses were made to infect the bacteria. Okay, so both of them were made to infect the bacteria. And what happened was that their findings would suggest that your protein in your experimental setup where protein coats were made or were done or were used, there is no radioactivity. Okay, there was no radioactivity inside the cell, but in that experimental setup where they used DNA, there was the presence of radioactivity. Okay, so they still confirmed, they confirmed further that it actually is DNA that transfers or that will transform your genetic information, your hereditary material from one organism to another, from one cell to another, not protein, not your RNA. Okay, so all these experiments have confirmed that it is DNA which is considered our blueprint of life, our genetic information that can be found, of course, in the nucleus of your eukaryotes, in the nucleoid region of your prokaryotic cells. Okay, now these two other people here, they gave us the DNA structure that we know now. Okay, the DNA structure, double helix structure, the DNA, and these were James Watson and Francis Crick. Okay, now, as we have already talked about, your other scientists have already confirmed that it is DNA information. If it is your DNA that is okay, it has all the characteristics that you can see whenever you are looking at yourself in the mirror. your characteristics are, of course, dictated by your DNA. Okay, it's not protein, it's not RNA, but it is the DNA, which is considered the genetic information that we find in our cells. Now, again, the two other people that we have here, Watson and Crick, they gave us the double helix structure of the DNA. But this actually was based on the findings of Rosalind Franklin and Maurice Wilkins. Okay, so these two were also the basis of the work of Watson and Crick. Let's take a look at your pictures here. Okay, so again, these are Watson and Crick, and they have given us the DNA structure of your cell, or your DNA, your double helix structure of your DNA. This was the product of the studies of Watson and Crick here. And as I have mentioned, their basis was actually the work of Rosalind Franklin. So this here was Rosalind Franklin, and she actually died because of ovarian cancer. And it was believed that her death was actually caused by her excessive exposure to X-rays, because he was actually studying X-rays. He studied the structure of your DNA, and so she died because of the exposure that she got from those X-rays. Okay, but because of her work, as it has become the basis of the work of Watson and Crick, we are able to know the DNA structure that we have now. Okay, and we know that your DNA is a double helix structure. Okay, we are going to your double helix structure, the DNA, in the next slide. Okay, so this here is your DNA. Now remember, when we studied your biomolecules, we mentioned there are 4 molecules that support life. Okay, you have your carbohydrates, you have your lipids, you have your proteins, you have your nucleic acid. And we know that your DNA here is a type of your nucleic acid. And so we know that the building blocks of your DNA are your nucleotides. Okay, we also know that your nucleotides consist of 3 parts, your 5 carbon sugar. In the case of your DNA here, we call this sugar your deoxyribose. Okay, now you also have your phosphate group. Now when you say phosphate, this is just your phosphorus, which is bonded to 4 atoms of oxygen. So usually we just use the chemical formula PO4. Okay, so 1 phosphate atom bonded to 4 atoms of oxygen. And of course, you also have your nitrogen bases. Okay, your nitrogen bases, these are the things that you find here, and we are going to talk about that later. Okay, so again, these are the 3 structures of your DNA. You have your 5 carbon sugar, which is your deoxyribose. You find it here. And then you have your phosphate group. You have 1 atom of phosphorus attached to 4 atoms of oxygen. And you also have your N bases. Okay, what are the different types of your N bases? We will study that in the following slides. Any questions so far? Any questions so far, people? Okay, now again, we've mentioned that the structure of your DNA is a double helix. Okay, it is your double helix, it has your sugar, it has your phosphate group, and it also has its N bases. Now, if you are looking at the structure of your DNA here, the double helix structure of your DNA, the handrails, the pink colored structure that we find here in our illustration, the handrails, if you consider your DNA like stairs, like your staircase, or like your ladder, the handrails are actually a combination of your sugar and your phosphate. Okay, so again, your sugar and your phosphate group can be found in the handrails of your DNA structure. Okay, so the pink color structure here, again, this is a combination of your deoxyribose sugar and your phosphate group. Now, your N bases can be found in the middle. Okay, the N bases can be found in the middle here, and this would form the rungs of your ladder. Okay, so again, if you are going to be asked, where can you find the ribose? Where can you find the sugar? Where can you find the phosphate group in your DNA structure? Again, that would be forming the handrails of your DNA complex. Okay, and the rungs, the steps here in the middle, those are actually the combinations of your N bases. Okay, so those are the N bases that you find in the middle. Now, some of these bases are held together at the center by either 2 or 3 hydrogen bonds, which among them are formed or are held by 2 bonds or by 3 bonds. I'm going to talk about that in the following slide. Okay, now looking at your nitrogen bases here, we actually have 4 different nitrogen bases in your DNA, okay? Remember, we've already mentioned before, you have 2 major types of your nitrogen bases. They can either be purines or pyrimidines, okay? When you say purines here, these are your N bases, your nitrogen bases that have double rings of carbon. Okay, they have 2 rings of carbon. So example for this here would be your adenine and your guanine. Okay, so adenine and guanine are 2 types of purines that can be found in your DNA. Now, your pyrimidine, on the other hand, this is an N base that only has a single ring of carbon. Okay, it only has 1 ring of carbon. And in your DNA, you see this as your cytosine and thymine. Your hint here is that the word pyrimidine has the letter Y, and your examples of your pyrimidines in your DNA are your cytosine and thymine. They also have the letter Y's in them. There is another type of pyrimidine as your N base, but this can only be found in RNA. Okay, this was part of your warm-up today. You were made to give some differences between your RNA and DNA. There's also a third type of your pyrimidine, your uracil, but this can only be found in your RNA. Okay, so we are not going to study this today because today's focus, of course, is your DNA. Any questions so far? Any questions, people? Okay, if you have no questions, let me move on. All right, now we've talked about your DNA and your nitrogen bases being in the middle of your DNA structure. Again, remember your sugar and your phosphates can be found as the handrails of your DNA molecule, okay? And the rungs in the middle, those are your N bases. Now, there is what we call a complementary base pairing rule. Okay, when you say complementary base pairing rule here, the pairing of your N bases are fixed. Okay, their pairings are fixed. That means your adenine would always be paired up with thymine and vice versa. If you see a thymine, okay, it would always be paired with your adenine. If you see a guanine as your N base, it would always be paired with cytosine. Okay, remember this. It's always going to be AT and GC, okay, or vice versa. It can be TA or AT. It can be GC pairing up with or CG, okay. Now, what's the difference between these two? If you try to take a look at your structure here, okay, looking at the structure that we have here, please pay attention. Again, your thymine would always be paired with adenine. Your cytosine would always be paired with guanine. Okay. Now, the difference is that in the number of your hydrogen bonds between these 2 pairings, your adenine and thymine are going to be bonded using just 2 hydrogen bonds. Okay. There's only 2 hydrogen bonds that are connecting your adenine and your thymine. Your cytosine and guanine, on the other hand, they are connected with 3 hydrogen bonds. Okay. So CG is connected with 3 hydrogen bonds, and AT are connected with just 2 hydrogen bonds. Can anyone tell us what the difference is now? Since there is a difference in the number of hydrogen bonds that connect them, what then would be the result? Anyone? Anyone who can tell us the result of this? What is the result of the difference in the number of hydrogen bonds that we see here? Okay, AT is connected by 2 hydrogen bonds, while CG or GC are connected by 3 hydrogen bonds. What do you think would be the difference then?
It would take more energy to break the hydrogen bonds of CG than AT.
Very good. Okay, so CG here, that's correct, Nasir. Good job. So CG here, again, would take a lot more energy to break, okay? Because it has 3 hydrogen bonds, it is going to take more energy, just what Nasir has said, rather than just the AT being broken, okay? So AT is going to take less energy than your CG because this here is connected by 3 hydrogen bonds. Okay. Now, another thing that you need to remember, aside from AT and GC being your complementary base pairing rule, is that you also have your Chargaff's rule. Okay. When you say Chargaff's rule here, the percentage of your adenine will always be equal to the percentage of your thymine. Okay. The percentage of your cytosine will always be equal to the percentage of your guanine. That means the number of thymine that you have in your DNA molecule would always be equal to the number of adenine that you have. Because remember, your thymine and your adenine would always be pairs. Okay? The number of guanine that you have would always be equal to the number of cytosine that you have. Because remember, they'd always be forming pairs. This might be asked in your benchmark on Monday. Say your benchmark on Monday would tell you there are 40 adenine in this DNA structure. Based on this, how many thymine do you think there will be? Okay, so of course, if there are 40 adenine, there's also going to be 40 thymine. Okay, because always, whatever is the number of adenine, that's always going to be equal to the number of thymine that you have. Whatever is the number of cytosine, that's always going to be equal to the number of guanine that you have. Okay, that's according to Chargaff's rule. All right, now this here is DNA replication. Please bear with me. This is very important. DNA replication is when the DNA is copied in a cell before it divides. We all know that DNA replication happens during the S phase of your interphase. Okay, you remember your S phase in your interphase. We said interphase is the longest part of your cell cycle. And out of the three stages there, your G1, your S, and your G2, S phase is the most important. Okay, S phase is the most important because DNA is replicated in your S phase. Chromosomes are replicated during your S phase. Okay, so DNA replication happens in the S phase of your interphase. Now, when this happens, when you try to replicate your DNA, out of one molecule of your DNA, out of the single molecule of DNA that you start with, you'd actually end up with two molecules of DNA. Okay, and these two molecules of DNA, if they have two strands, of course, they are going to have two strands of DNA molecule, right? In their structure, there's going to be two strands. Out of these two strands, one strand would be coming from the old DNA molecule, and the other strand would be a new molecule, okay, or a new DNA strand. And so we call this a semi-conservative process. Okay, semi-conservative, because your two DNA molecules that are going to be produced after one round of replication, it's going to have one strand from the old DNA molecule and one new strand. Okay, so again, we use the term semi-conservative. Okay, now let's take a look at the different stages or different steps in your DNA replication. First step, you have the enzyme called DNA helicase. And what it will do is that it is going to unwind your DNA structure. Okay, remember your DNA molecule is a double helix. Now your DNA helicase is going to unwind that double helix. Okay, because remember, we are going to make copies of your DNA. And it's very difficult if your structure is a double helix. So what it does, your DNA helicase, it's going to unwind the structure of your DNA. Okay, now your nucleotide bases, the bases of your nitrogen here, are going to be separated, and you'd call this part your replication fork. Okay, that's the replication fork. That is where your DNA replication is going to start. Okay, any questions, people? So again, that is the first part of your DNA replication. DNA helicase is going to unwind the double helix structure of your DNA. Okay, and your N bases are going to be supplied in what you call the replication fork. Now, after this, what happens is that your DNA polymerase, this is another type of enzyme. DNA polymerase would move along each DNA strand, and it is going to add complementary nucleotides. Okay, so remember, adenine always pairs up with thymine, cytosine always pairs up with guanine. Okay, so whenever your DNA polymerase would see an adenine, for example, it is going to pair that up with a letter T, or your thymine. If it says letter C, your cytosine, it is going to pair that up with guanine, your letter G. Okay, remember, A and T are always together, C and G are always together. And so in the end, there's going to be two molecules of DNA formed. Okay, and remember, this is the DNA molecule that we started with. This is going to be to get unwind by your DNA helicase. Then your DNA polymerase is going to supply the N bases there. Okay, so in the end, after one round of DNA replication, you have two molecules of DNA. Now, as you can see in our illustration here, these two molecules of DNA has one strand coming from the old DNA structure, and also has one strand which is entirely new. Okay, so we call this process again a semi-conservative process. Okay, it is called semi-conservative because one strand of the DNA molecule came from the old DNA molecule, and one strand, the other strand, is entirely new. Okay, any questions, people? so far, people? Any questions? All right, now say let's have our practice here. Okay, take a look at your slide, take a look at your screen. We are going to fill the nucleotides with the proper letter. Okay, so you have your DNA molecule here, and you have the following N bases. Okay, the first base that we have here on top is a letter A. Can anyone tell me what letter would complete this base here, this complementary base pairing here? What letter should we write here so that we can complete the base pair? Okay, Jason says T, Donovan says T, that's correct, very good. Okay, so that's T, good job. Okay, so again, if you have an A here, that's your adenine, it is going to be paired with your letter T, that's thymine. Because remember, A and T are always together. Now we go to the second rung here, you have your T. What should we write here so that we can complete your base pair? Very good, very good. Good job. Thank you, everyone. Okay, congratulations. That's correct. Okay, so again, this here would be a letter A. That's a letter A because, of course, the pair of your thymine, that would always be letter A, that's adenine. Okay, remember, A and T would always be together. T and A would always be together. Okay, so AT or TA. Now, next rung, you have G. That's guanine. What should we write here to complete your base pair rule? Okay, good job. Thank you. Okay, congratulations. That's correct. So that means this should be letter C here because that is going to complete your base pairing here. Okay, based on our complementary base pairing rule. Now, what about your letter C? Your cytosine. What's going to be paired with your cytosine? That's letter G. That's guanine. Good job. Okay, so that is correct. Very good. Now, let's take a look at our next slide here. Okay, now this here is more complicated than the one that we just had. We are going to replicate this DNA molecule. Okay, say you have this DNA molecule. Those are the strands of your original DNA molecule. Okay, now this has been acted upon by your enzyme DNA helicase, and it got unwound. Okay, so it has been unwound by your DNA helicase. So now you have these two original strands here. Okay, one strand here that was the upper strand from your original DNA molecule, and you also have the lower strand here found at the bottom. Okay, now I'd like you to supply the new strand for the first original strand that we have here, and also supply the new strand for the lower part that we have at the bottom. All right, so again, going back to our activity here. Okay, you have your original DNA molecule with these strands. Okay, the N bases, the sequence of nucleotides are found there. And then it has been unwound by your DNA helicase. Now we have the original strand separated. So what you do is you just look at the lower portion that you have here, and that actually becomes the new strand. Okay, that would be equal to your new strand because remember, we've got this from this original DNA molecule, and we know that these are already complementary. Okay, so to complete your new strand here, that should be the same as the original strand that you have here at the bottom. Okay, so that would just be TAGGCT. Okay, so TA, then GGCT, and so on. Okay, now to complete your new strand here at the bottom, that should just be equal to your original strand here, the one that we find on the top. Okay, so the bottom part here would just be ATCCGA. That's correct. Okay, that's correct. Okay, good job, Angela and Adriana. Okay, you see that? So again, for you to be able to write the new strand, the challenge, it's going to be more challenging if you are just given one strand. Okay, if you don't have this as your basis, that would happen when we study the RNA. But here, since you have the complementary basis, what you need to do is just copy this. So just copy this here, and this one at on top, you just copy this here as your new strand. Okay, now remember, at the end of your replication, you produce two molecules of DNA, and both of them are semi-conservative. They're semi-conservative because they have an old strand and they also have a new strand. Okay, any questions? Any questions, people? All right, now I believe this is the last slide that I have before I give you your independent work. Now, some errors, sometimes 1 out of a billion can happen. One error out of billion can happen. happen. And this is corrected by your DNA polymerase. Remember, your DNA polymerase is also the one that supplies your nitrogen, your nucleotide sequence. Your DNA polymerase is also the one that proofreads the DNA and repairs errors. Now, whenever there is an error, of course, you'd call that a mutation. Okay, any error in your DNA structure, any error in the chromosome, you'd call that a mutation. And sometimes this mutation can be harmful. We've seen that yesterday in your trisomy 21 in people who suffer from mongoloid, those people who are mongoloid. There can also be some beneficial mutations. Sometimes they can also have no effect. Okay, so again, any change, any error in your chromosome, any error in your DNA, we call this mutation. Okay, and again, your DNA polymerase, aside from supplying the nucleotides, the N bases during DNA replication, it also proofreads your DNA.